What? Yes. 1. Yeah, it should be. Did people get the lecture slides? Because were they available? Yeah. Okay. So they should be there.
Hi.
Hello.
Joel emailed me, and he's saying that he needs start invitation so that I would use the answer and do. I thought talking to him.
Yeah, I mean, we just technically we're supposed to get.
Yeah, well, here's the document.
Okay. Sorry about that, but yeah, that'll be fine.
Okay.
Do you need this back, or?
I mean, I guess. Yeah, I probably do.
Maybe I'll make a copy of it and get it back to you. And I'll send you your calculated grade.
Okay, thank you so much.
All right. Gonna get started. Remember me? I was here a while ago. Hi. Today we are going to wrap up the course. And what I want to do in lecture is two main things here. Is that too loud or no? No. Two main things here. One is to try and connect together many of the different threads in the course with a story, or maybe two stories, about a biological system. And also, it's a biological system that I've been involved in studying, so it's also a little story about detective work in understanding this biological system. So the previous lecture was things I don't know anything about, as people who have asked me questions in office hours now. And then, of course, after this is the final. For Section A, last names A through G will be in 3 Kleber. Names H through Z will be in Rock Hall. And again, bring a pen, a number 2 pencil, and a photo ID. In addition, if you haven't done this already, we really, really would like to get everyone to fill out this online evaluation system. We definitely use the feedback to try and understand the good and bad parts of the course, and you should be able to do that online. Yeah. What? A TA? Yeah. I'm not sure. If there is a TA, then it would be your specific. All right, we will have to try and fix that. You don't need, and are we on there? Interesting. Yeah. Yeah, just don't fill out the one for that person. I mean, it probably was supposed to be for Susan Keene, but people would have been confused. And I will send around an email. I'll try and get it fixed and send around an email. I didn't notice that when I looked at it. All right, so here's your first clicker question of the day. I hope you get it right. There have been people in the past who have gotten it wrong. How much time do you need for this? All right, another, for all you stragglers out there, another 5 seconds for this very hard clicker question. All right, I'm going to stop it. Excellent. I'm very proud of everyone. All right, so now on to the actual story. So what I want to tell you about is a story that in many ways relates to Davis in our area. I got involved in this story when I was living in Maryland, actually, so it was before I was out here in lovely, nearby lovely wine country. So if you're driving around anywhere in any wine growing region and you see some nice setting like this with a vineyard looking all pretty and happy, and everybody's probably pretty happy with that. If you're looking closely at a vineyard and you see something like this, that's a bad sign. So the leaves on the left and the whole trees, whole vines over there are not looking particularly healthy. There are many different things that can go wrong with grapevines, but this is evidence of something that's really, really bad. It's a disease called Pierce's disease, and if you find it in a vineyard and the authorities know about it, they're probably going to want you to destroy the entire vineyard. It is very contagious, and it ends up eventually killing the plants, or at least making them not produce grapes or anything that you would want to consume. It's a major economic threat to grape-growing regions. Pierce's disease actually also occurs in some other plants, but we're going to focus on grapes today. It's endemic in the southeastern United States, and it can be pretty devastating when it comes into a new area and infects the plants that are there. It's caused by a bacterium called Xylem fastidiosa. This bacterium infects the xylem circulatory system in plants. Do you remember what the xylem circulatory system is? What's the difference between xylem and phloem? Yeah, so xylem is the circulatory system for moving water around. Phloem is the one for moving nutrients around, such as those made by the leaves and the chloroplasts. Pierce's disease and the bacterium that causes it, Xylella, is a topic of an enormous amount of research because of the economic damage that it can cause in various agricultural regions. If you're interested in this type of thing, there are quite a few people at UC Davis and at other UC campuses who specialize in studying this bacterium or the disease that it causes. And again, we just sort of talked about this, but basically it's an infection that occurs in the xylem circulatory system. It's not clear exactly what the mechanism is by which it eventually damages plants. It is probably connected to, in essence, clogging up the xylem circulatory system with overgrowth of bacteria. They may also secrete toxins inside the circulatory system that damage different parts of the plants. So this interaction between Xylella bacterium and the grapes is a type of symbiosis. What kind of symbiosis is it? Right, so it's a form of parasitism. These organisms are tightly affiliated with each other, so it's a symbiosis, and Xylella benefits, the grapes do not, so it's a form of parasitism between a bacterium and a plant. If you want to study the biology of this bacterium, there are many things you can do. We talked about the general pattern with which you could go about trying to study microbes. One of the first things you can do is look at what's going on in the field. This is from my general slide. On average, for bacterial studies, the appearance is not overly useful to understand what's going on in the field. There's an exception in this case, which is that if you found a plant infected with a bacterium and you didn't know what that bacterium was, you couldn't tell what it was probably from the appearance. But you can tell a lot about the infection itself because you can see the Xylella bacterium in different parts of the xylem circulatory system. So field observations are very useful. They're not useful for identifying which bacteria might be involved in this infection. So when this was first discovered, people found the leaves browning. They went in and looked at the different parts of the plant, and they cut it open, and they found these large, clusters of bacterial-like cells growing inside the xylem circulatory system. The next thing to try to do is to grow the organisms in the laboratory, to culture them. And it turns out that Xylella fastidiosa is culturable. You can work with it in the laboratory. And so that opens up a whole massive window of microbiological techniques, genetic techniques, genomic techniques, et cetera, to try and figure out what's going on with this organism, how does it damage plants, what might kill this organism, and other factors. Eventually, people working on this organism did ribosomal RNA sequencing that told them that it was this Xylella organism, and then in the last few years, people have sequenced the whole genome of the Xylella bacterium and used that genome to make predictions about the biology of the organism. We're not going to talk much about Xylella, but the genome was very useful in better understanding how it might interact with plant systems and what it might be doing as part of its infectivity. We haven't talked a lot about infectious diseases in plants. We've talked a little bit about some of them in animals. So how is an infectious disease going to get between, let's say I have the flu. How's the flu going to get between me and people sitting right here in the front row? In the air, or if I rub my hand and I go and shake hands with someone, I can transmit an infectious disease because there's this thing about animals where we move, right? Professor Wainwright told us a little bit about that. They actually have some sort of muscle or something like that they can move around. I don't know anything about it, but yeah, real animals, that's right. And so I'm going to show you a very brief little video here. This is grape plants, time-lapse video. They're not going anywhere. They're growing, but an infectious disease in grapes has an awkward time in transmitting itself to other grape plants if it's going to depend upon sort of the leaves banging into each other or some other type of interaction. And what I'm not sure if the audio is going to work on this. There's a little video here that talks about what's going on with this Pierce's disease, how it is transmitted between different grape plants, and a little bit about the agent that does the transmission. It was made in like the 1840s, but that's it.
state's abundant sunshine and mild Mediterranean climate make it an ideal location for growing grapes. And the wines that come from these vineyards are prized the world over. A new invader that has the potential to cause serious damage now threatens the state's wine, raisin, and table grape industries. It's already helped decimate the grape industries in Florida and Texas, and now it is California's turn to deal with this menace. This new intruder is an insect known as the glassy-winged sharpshooter. A native of the southeastern United States, scientists believe it was brought into Southern California in 1989 through infected nursery plants. Since its introduction, it has spread rapidly. So far, it's been found in 15 California counties, mainly in Southern California and the southern San Joaquin Valley.
Well, we're here in a Ventura County lemon orchard. An orchard that's about 5 years old. And the concern here locally with agriculture has been a relatively new introduced pest species, the glassy-winged sharpshooter. It occurred here in Ventura County about 10 years ago when we first saw it, and it has developed its populations over this last 10 years and has reached some very, very large levels to where in young lemon trees such as this one here, we can have populations of 50 to 100 or more insects per tree.
The adult insect is almost half an inch in length and is dark brown to black in color with a lighter underside. The upper part of the glassy-winged sharpshooter's head is marked with ivory or yellow spots that are visible when viewed up close. The insect gets its name from the semi-transparent wings on its back that are laced with reddish veins, and the characteristic side-to-side motion it sometimes exhibits, similar to a sharpshooter aiming at a target. Glassy-winged sharpshooters are voracious feeders, and they are not picky. They're known to feed on over 130 different types of plants. These pests usually feed on the stem of a plant rather than the leaves. They have remarkably strong mouth parts that are able to penetrate down inside the stem to the part of the plant that conducts water from the roots to the leaves. This tissue is known as the xylem. Because the fluid within the xylem is very low in nutrients, the glassy-winged sharpshooter needs to consume large amounts of it in order to get enough energy to live and grow. When feeding, it excretes this liquid as a stream of small droplets that rain down to surfaces below after filtering out essential nutrients. In areas where the glassy-winged sharpshooter populations are very large, a mist made up of these droplets can actually be seen. As this watery excrement dries, it leaves behind a white residue. The real threat from the glassy-winged sharpshooter is not from the damage it does directly, but from a disease that it helps to spread. The glassy-winged sharpshooter can carry a bacteria that causes Pearson's disease.
All right. This insect, the glassy-winged sharpshooter, is an obligate xylem feeder. It lives feeding off of xylem of different plants. One of the plants that it can feed on is grapes, and if it feeds on a grape plant that is infected with xylella, it can take up the xylella into its system and then eventually transmit the xylella to another uninfected plant. As an aside, it is the only animal listed by Department of Homeland Security as a potential bioterrorism agent. People are if you had some infected glassy-winged sharpshooters and you introduced them into Napa, that would cause billions of dollars of economic damage. So people got a little freaked out about this maybe 10 years ago. I haven't seen as much about it. But it basically works like mosquitoes work in transmitting diseases in animals and in people. So mosquitoes feed on blood. If your blood is infected with something like Plasmodium, the causative agent of malaria, the mosquito can pick it up and then eventually go transmit that disease agent, again like Plasmodium, to another uninfected individual. So the sharpshooter is a vector for this disease. And even though there are many other sap-feeding insects that will feed on grapes, the glassy wing sharpshooter is particularly good at spreading this disease. And it's why if you drive around Napa, there are pictures of this insect up on signs saying, if you spot this insect, call the US Department of Agriculture. It's a real major worry in regions of the country where it has not shown up yet. Pierce's disease devastated the grapes growing in many other parts of the country. The people in Northern California would very much like that not to happen. And this is just to gross you out. This is a tree that is infested with glassy wing sharpshooters, millions of them. Watch what's coming down from the tree. That's the rain coming out of their backside. They're feeding so much that you just collect this sort of mist all around the tree, and eventually it basically starts to rain glassy wing sharpshooter pea. They said, there's audio on this that I cut out, they said that these were not paid models, these were just random people in the street that they were videoing, looking up at the raining tree. So there is another symbiosis here between now the Xylella and the sharpshooter. They have this sort of tight association, the sharpshooter distributing the Xylella between organisms. Without knowing anything else about this, many people have thought that this type of interaction is some form of commensalism. Sharpshooter is just serving as a vector. If the Xylella is there, it gets transmitted. If not, the sharpshooter doesn't really care. However, in the last maybe 10 or so years, many people have been suggesting that, in fact, one of the reasons the sharpshooter may be such a good vector for transmitting this disease is that the Xylella might actually be doing something for the sharpshooter when it is inside the sharpshooter. So some people have suggested that this is another mutualistic interaction, this time between the sharpshooter and the disease that it transmits in the grapes. Okay, so going back to what the sharpshooter is actually doing, so you saw in that little video, it's feeding on Phloem Sap, the Xylem circulatory system. In most, basically all plants, what's in the Xylem circulatory system is basically mineral water. There's a little bit of organic compounds in there, enough such that the sharpshooters, which are obligate Xylem feeders, that's all they're eating, they can basically filter out a little bit of sugar and a little bit of other organic compounds out of this xylem, enough to get enough energy to grow. And again, they have to filter out so much that they're basically constantly streaming water. They're like a straw with a little filter on it that's capturing organic compounds out of the fluids and a few other minerals and other things. Phloem sap, on the other hand, is something that in general should make you a little bit happier if you were feeding on it. So you have this insect that's feeding on the Xylem circulatory system, which is pretty weird because of the low nutrient content. What about phloem sap? Phloem sap is actually filled with all sorts of lots of sugar coming from the leaves where photosynthesis is happening. We didn't talk about it in that much detail, but in the phloem, there's also distribution of vitamins and amino acids and other compounds that are being synthesized in various parts of the plant. And so you might think that phloem sap would be a better target for consumption, and in fact, it is. So there are many, many insects that are obligate phloem feeders. Those insects do pretty well. So this is an aphid. If you go out and look at rose bushes at particular times of the year, you can see that they're completely covered in layer after layer after layer of aphids feeding on some of the newly made parts of the plant and getting some of the yummiest phloem out of them. They have the same thing going on. It's not quite as fast as with the sharpshooter because they don't need to drink quite as much of the liquid, but they are still basically filter feeders, drinking the phloem circulatory fluid and then filtering out what they need out of it, and they have to get rid of the liquid out of that. Aphids and their relatives are incredibly successful organisms. They've undergone a massive amount of diversification. There are at least 4,000 species, and you can find them basically, everywhere where there are plants. They're just voracious eaters. They reproduce. Many of them reproduce by parthenogenesis, where a female can just basically clone herself, and they can reproduce in massive numbers in very short periods of time. Aphids are doing really well. And in part, that relates to the food that they're getting. It's sort of the difference between drinking mineral water and drinking, maybe some type of smoothie of some kind. Now, plants are evolving all the time in response to threats from various predators and organisms that are trying to eat the different parts of the plant. So there are many things that if you think about it, in the past when plants first started to colonize the land and when they started to grow and they started to have these complicated circulatory systems distributing different types of compounds, and then some insects came along and evolved the ability to get into the phloem circulatory system and start drinking that. Plants most likely would be under strong natural selection to have some changes that would prevent the sap-feeding insects from doing this. Sorry, I don't have I may not have a slide on that. But so plants could put out waxy coatings on different parts of their stems to try and make it harder for things to penetrate. They can produce secondary metabolites, toxins, which we see in lots of different plants that will kill organisms if they're eating from the sap. They have thorns. They have all sorts of means to protect themselves from various threats. Aphids are able to get around most of those threats and still eat from the phloem circulatory system. But one thing that plants have also evolved to do is that they have generally removed certain nutrients from the phloem circulatory system that animals need. So animals, there's a set of nutrients that animals need to get from their heterotrophic diet. They need to consume certain things. We don't fix our own nitrogen, for example, so we need to get nitrogen products from our diet. We can't, even if we get simple nitrogen compounds, we can't make all the different amino acids ourselves, and this is true for all animals. There's a set of amino acids known as essential amino acids. They're known as essential because we have to get them from our diet. And so plants, over evolutionary time, have picked up on this, and they have removed the essential amino acids from their circulatory system to make it more difficult for an animal to live off of eating or drinking from their system. And so what is an animal going to do in that context? What are the aphids going to do, for example? Well, they could eat other things. So it turns out there are many organisms that some of the time will drink phloem sap, but they don't depend on it. They'll get nutrients from other sources. Aphids don't do that. They're obligate phloem feeders. They can't do anything else. So the aphids could evolve metabolic pathways to synthesize all those missing essential amino acids. On average, animals aren't very good at inventing these new complicated metabolic pathways. And even if they were, it turns out that in most cases it's easier for these animals to engage in some type of symbiosis with another organism that can make what they're not able to get from their diet. And that's what happens in aphids. So what aphids do is, I'm going to tell you about this little story about what aphids do. Are there any questions about what we've been talking about so far before I get into what's going on in aphids, and then we'll come back to sharpshooters? Okay, so go back about 30 years, maybe even 40 years. Two professors at UC Davis, Paul and Linda Baumann, have been studying sap-feeding insects for many, many years. They retired a couple of years ago, but they were here for a long time studying sap-feeding insects. And one of the things that they did is if you dissect sap-feeding insects and you look inside their gut, you see specialized organs inside their gut that are chock full of things that look like bacteria. Again, you can't tell exactly what they are by looking at their appearance in a microscope. So if you look at these images that you see, they certainly look like bacteria, but they could be something else. They could be weird mitochondria. They could be archaea. They could be weird parasitic protists. Wasn't exactly clear. So what do you want to know? You want to know who they are and what are they doing. So the Baumans and colleagues of theirs, they do the field observations. The appearance didn't tell them what they were doing. So they spent a lot of time trying to grow them in the laboratory with no success. These symbionts that live inside most of these aphids and sap-feeding insects, no one has ever been able to grow them in the lab. So what do you do instead? If you can't grow them and the appearance isn't useful, anyone? I can't hear what. I couldn't hear it, but I'm just going to assume everybody knows. So you look at their DNA, right? So that's the culture-independent studies. If you can't grow things and if appearance isn't useful, you go and you look at their DNA, and that's exactly what the Baumans did. They did ribosomal RNA PCR amplification to pull out the ribosomal RNA genes from these tissues and look at what kind of organisms those little bacterial-like cells were by building sequence alignments, making a data matrix, and building an evolutionary tree, right? You remember all this? You go to the sample, you extract DNA, you make lots and lots of copies of the ribosomal RNA genes from the sample, you read the sequence of those, you compare the sequences to sequences from organisms that you know something about, you build a data matrix, and then you build an evolutionary tree. You look where your new ribosomal RNA shows up in the evolutionary tree of those organisms, and this is what they got. Here's an evolutionary tree where they were comparing their organisms to other members of the Proteobacteria phylum. The Proteobacteria phylum is one of those 6 major groups of bacteria that we talked about a long time ago. It includes E. coli, for example. And what they found actually was that the symbionts of these aphids, these things inside the gut tissue, were very close relatives of E. coli. They're gamma subgroup of the Proteobacteria. So that doesn't tell you exactly what they do, but it tells you, it guides you in how you can then do experiments to try and better understand them. Because if these were closely related to halophilic archaea, the type of experiments that you would do are very different than knowing that they're closely related to the model bacterium, Escherichia coli. So they spent a lot of time then using this information to better understand the biology of those symbionts. Eventually, I'm compressing 20 years of work into a couple of slides. Eventually, to get a full understanding of what these bacterial symbionts were doing, another group went out, extracted DNA, and read the entire genome sequence of these organisms. So even though they couldn't grow them in the lab, they could get out enough DNA from them to piece back together the complete genome sequence of these symbionts living inside the aphids. And then you take that genome data and you make predictions about the biology of that organism by comparing each individual gene to genes from other organisms. This worked out really well because these bacteria were very closely related to E. coli, and we know an enormous amount about the genes in E. coli. And each of the genes in this symbiont genome, it became known as Buchnera, that's what the symbionts of aphids are called, has a match in the Escherichia coli genome. So it was pretty easy to make predictions about the biology of these bacteria living inside the guts of these aphids. And to make a long story short, what these bacteria are doing is synthesizing the essential amino acids that are missing from the aphid diet. They basically have a set of genes for running the core functions of a bacterium, like replication, transcription, and translation, and the rest of the genome is for making essential amino acids for their host. That's what they do. So what type of interaction is this between the bacteria, the Buchnera bacteria, and the aphids? Is it parasitism? Mutualism? So this is a mutualistic interaction where the host is providing sugars, actually, for the symbionts, and also giving them nitrogen that it's getting out of the circulatory system of plants. And then the symbionts are making the essential amino acids missing from the diet of the aphids. This is an amazing strategy. It now means that aphids can live off of incredibly nutrient-poor fluids, and it is probably responsible, or at least partially responsible, for the massive diversification that is seen in aphids and their success. They have basically a little food producer that they carry around with them so that they don't need to get amino acids from their diet. Okay, so now back to the sharpshooter story. Aphids have it lucky. They're drinking phloem. They're drinking, some sugary, organic compound-rich food. The sharpshooters are drinking xylem, which is really nutrient-poor, and plants have basically stripped almost everything out of most of the xylem except water and some minerals. So it's not only missing the essential amino acids, it's also missing all the vitamins that we animals also don't make that we get from plants. And it has almost no carbon skeletons with which to then build things. So that's why the sharpshooters have to pump so much fluid through them. They're basically collecting tiny amounts of sugar and maybe a few other organic compounds that they can get out of the xylem, but they're not getting vitamins, cofactors, or the essential amino acids. So let's go back to the glassy-winged sharpshooter. If we want to understand the biology of this organism and maybe prevent it from spreading Pierce's disease, it would be good to know how it is able to feed on xylem without dying, basically. Xylem-feeding insects are also very successful, just like phloem-feeding insects. The sharpshooter is a close relative of cicadas, which you may be more familiar with, that are pretty successful. There are quite a few species in this sharpshooter cicada relative group. So same issue comes up with the sharpshooters. What can they do in response to drinking this incredibly nutrient-poor fluid? They can eat other things. Many organisms eat or drink xylem but don't drink it solely. Sharpshooters are obligate xylem feeders, so they're not able to get anything else. Animals aren't very good at evolving novel metabolic pathways, so they can't do that. So again, most likely what they're doing is they have some type of symbiont that they're leveraging to make the things they're missing in their diet. Now comes in the second part of the story, which is a woman named Nancy Moran, who is now a professor at University of Texas, was interested in evolution of symbioses, and she read some papers by Paul Baumann and Linda Baumann about xylem-feeding insects that also appeared to have bacterial-like colonies in their gut systems. This is a different type of sharpshooter, not the glassy-winged sharpshooter. But you see these whole organs, they're called bacteriomes, and they had shown that there were bacterial-like things here. Nancy then collaborated with Paul Baumann to try and understand what these symbionts were and what they were doing. This is Nancy Moran. She's one of the most brilliant people on the planet. MacArthur winner, Japan Prize winner, other amazing things. She got interested in these symbionts. What do you do to figure out what the symbionts are doing? Same thing. You can look at them in the microscope. Just like with the symbionts of aphids, the appearance was not particularly informative as to what was inside these tissues here. So what do you do next? You try and grow them in the laboratory. None of them could be grown in the laboratory. What do you do instead? You look at their DNA. So Nancy collaborated with Paul Baumann to look at the DNA of these sharpshooter symbionts and eventually started doing work on them on her own at her lab at University of Arizona. You go to the tissue, you extract DNA, you run ribosomal RNA PCR, starting to sound familiar. You sequence the ribosomal RNA genes, you build a sequence alignment, and then you build an evolutionary tree, and that tells you what the organisms are. Here's an evolutionary tree showing what this organism is inside the glassy-winged sharpshooter. She named it after Paul Baumann, so it's Baumania cicadolinicola for cicada group. It's a close relative of the Buchnera symbionts of aphids. That's again in this group that's very closely related to E. coli, so they're proteobacteria, closely related to the symbionts of aphids. Based upon this, it seems reasonable to hypothesize that maybe these symbionts are making amino acids for their host, just like the Buchnera symbionts are of aphids. So figuring out what they're doing, if you can't grow them in the laboratory, one of the best ways to do this is to sequence the genome. Amazingly for me, Nancy came to me to help do this. I was working at this big genome center. We had interacted for many years over studying actually aphid symbionts in a different situation, and she said, would you like to put in a grant proposal with me to sequence the genome of the sharpshooter symbionts? And I pretty much said, I would do anything you ask me to do. We got a grant. Back in the day, this was many years ago, the total cost for sequencing the genome of this symbiont of the sharpshooter was about $100,000. We can now do it in the lab at UC Davis for about $50. But it was still fun. And so we sequenced the genome with her. She extracted DNA out of these samples, FedExed the DNA to us at this genome center that I worked at in Maryland. We read individual reads. We eventually stitched back together the whole genome because it's a close relative of E. coli. We could predict the biology of the organism really well. And we were sort of sitting there drinking our coffee or something like that, scanning through the data, getting ready to find all the essential amino acid synthesis genes. Here's a map of all the predicted metabolic pathways in this organism. And surprisingly, we found all these pathways for synthesizing vitamins and cofactors. That makes sense. That's missing from Xylem, from the Xylem circulatory system. So that was really cool. This was the first example of one of these insect symbionts making vitamins and cofactors. But there were no pathways in the genome for synthesizing essential amino acids. We were vexed. We were very confused about this. It seemed like they had to be there. The symbionts had to be making essential amino acids for the host because they weren't getting it from their diet. So then Nancy told us that she had some new data that she hadn't yet published. They were just writing up a paper on it. She hadn't, in fact, told us about this data where they had gone to this sample, done the ribosomal RNA PCR, and there actually was a second ribosomal RNA gene that came out when they dissected these tissues, less abundant than the first. They paid attention to the first one because it came from an organism that was very closely related to the Buchnera symbionts of aphids. She kind of had ignored this one, but now was paying more attention to it. It's a second symbiont. It's actually eventually got named Solsia mulleri. It's in a group that we never really talked about, but I mentioned briefly when we talked about the human microbiome. The Bacteroides group is a group of bacteria that are very frequently found in association with diverse animals as mutualistic symbionts. So here's this other bacteria in the sample. It turns out this is an fluorescent microscope image from the tissue where these symbionts are. In green are the Baumania. In red are the Solsia. The Solsia cells are enormous. They're very, very big bacterial cells, and when we had sequenced the genome of this Baumania, we wanted to get rid of the sharpshooter cells, so we filtered out big cells from our DNA sample, and we accidentally got rid of the Solsia DNA at the same time. Eventually, we went back to other samples, did some genome sequencing. A postdoc in Nancy's lab, John McCutcheon, did some more genome sequencing, and eventually the entire genome of this organism, Solsia, was stitched back together, and it's an essential amino acid producing machine. So here you have this dual symbiosis. The sharpshooter can't get vitamins and cofactors or essential amino acids from its diet. And the way it gets them is it feeds sugar to two different bacterial types that live in these special organs in its gut. The Baumania makes vitamins and cofactors. The Solsia makes essential amino acids. And if you look, I don't have the figures to show you this, if you look at the diversity of sharpshooters and compare them all to each other, these two symbiont groups have been associated with sharpshooters since the origin of the sharpshooter group. In fact, since the origin of the whole clade that includes cicadas, some 100 million plus years ago, this symbiosis evolved, and those symbionts are now maternally transmitted from parent to offspring, just like mitochondria are. They are co-evolving with their hosts, in essence, for hundreds of millions of years, these obligate intracellular symbionts that are starting to resemble organelles like the mitochondria. So this symbiosis with the sharpshooter sort of obviously is a mutualistic interaction. And understanding of this interaction has now been leveraged by some of the people who are trying to, again, prevent the sharpshooter from transmitting Pierce's disease in grapes, and also it transmits other pathogens in other organisms. So understanding what its Achilles heel is, basically, are these bacterial symbionts that it needs to live and feed on its diet is really important for designing ways to try and get rid of it. And hopefully that information will contribute a little bit to preventing, understanding, and stopping Pierce's disease. So are there any questions about the sharpshooter system before I switch at the end to just talk about one other symbiosis? It's this amazing example of three-way mutualistic symbiosis where each of the partners is making something that the other can't do. Is that a question or? No. So, was there a question? No? Okay. So we've seen a lot of examples of mutualistic interactions in the class. Most of them have involved a single organism interacting with another organism, but we've seen a few that are more than that. But for example, the eukaryotic organelles are a mutualistic interaction that evolved a very long time ago, the plastids and the mitochondria. The secondary symbiosis, where one eukaryote brought in another eukaryote, is another form of likely mutualistic interaction. Lichens have this interaction between fungi and photosynthetic microbes. Coral and dinoflagellates we've heard about, the ant fungus farming system, legumes and nitrogen-fixing bacteria. We haven't really talked about it much, but there are many organisms that eat cellulose-containing material as all of their diet. Ruminants, for example, they can't digest the cellulose themselves, and most of these organisms have microbes in their gut that digest the cellulose for them. And the last thing I want to talk about is this one example which Professor Wainwright mentioned briefly before. Just want to tell you a little bit more about it, are these tube worms and chemosynthetic bacteria. So I want to tell you this because it's just an amazing story about the diversity of life, but also a great example of mutualistic symbioses. So in 1977, a bunch of geologists were going on a surface boat near the Galapagos Islands in a place called the Galapagos Ridge. And they had a trawl basically behind the boat that had a temperature sensor and a camera on it. And they were trying to look for evidence for seafloor spreading, for cracks in the seafloor where hot water might be coming out. And they found a region where there was a spike in temperature based upon this probe. And then they sent down 3 people in the Alvin submarine, this deep-sea submersible, down to the bottom of the ocean where this temperature spike had been recorded. And what they found in that place where they went was amazing. It was the second highest density of biomass next to tropical rainforests. They found a site now known as Hydrothermal Vents where there were these cracks in the seafloor, energy-rich compounds coming out, and an entire ecosystem that was formed, that was there with, piles and piles of organisms, including these giant tube worms, which became sort of the landmark organisms from these systems. Oh, I didn't even know there was audio here. They're just amazing. Some of them can get to be about 6 feet tall. They have this giant red plume that sticks out of the top of them, out of the top of their tube. And when these were first found, the scientists that found them were very vexed. They didn't understand what they were doing. They have no mouth. They have no digestive system. They're this very strange, obviously a worm-like organism, but they're missing many of the key parts of what you think would be essential for a worm. One of the scientists who was working on these from the Smithsonian gave a talk at Woods Hole where a graduate student, Colleen Cavanaugh, this is a recent picture of her, but when she was a graduate student at Woods Hole, she was actually studying chelicerates. She was studying horseshoe crabs, and she heard this talk about tube worms from this person from the Smithsonian, and he said, we don't understand what they're doing. They have no mouth, no digestive system. They have this weird liver-like organ, and inside that liver-like organ, we see globules of elemental sulfur. And she basically literally got up in the talk and said, no, they're going to have bacterial symbionts that use the sulfur inside that organ, and they're going to function like plants with chemosynthetic bacteria living inside of them. And he said something to the effect of, silly girl. But eventually she got samples of the tube worms, did electron microscopy. It's a pure culture of bacteria inside this giant liver-like organ. And then she did ribosomal RNA sequencing with Norm Pace, actually, the person who pioneered the whole environmental RNA sequencing, showed that these were, in fact, bacteria living in there. As a graduate student, she had a paper in the very prestigious journal Nature, and then a paper in Science that I think was on the cover of Science by not paying attention to this scientist who thought that she was wrong. And it turns out this is an amazing system. Inside these tube worms, there are bacteria that the tube worm brings in hydrogen sulfide, carbon dioxide for the bacteria. And the bacteria are chemo-autotrophic. They fix the carbon using the energy and electrons from hydrogen sulfide, and then they feed the worm just like chloroplasts feed plants. And this entire ecosystem in the bottom of the ocean is based upon some form of chemo-autotrophy, either with free-living microbes or microbes that live inside these tube worms. It turns out you can find microbes living inside clams in the deep sea. This is a giant clam called Calyptogena. You can find them in little clams that live in the coastal environments. So it's not just in the deep sea. This is a clam called Solamaya. You can find them in snails in various deep sea environments. And this is all this amazing, relatively newly discovered system where the basis of life is not light energy. It is chemical energy driven by bacteria, archaea, and then symbiotic bacteria living inside these animals. So these are ecosystems that do not need light. And the reason what I want to leave you with here is Colleen Cavanaugh was my undergraduate advisor. She's the reason I got interested in microbes and weird deep sea systems. That's me collecting tube worm samples from the back of the Alvin submarine after getting interested in this with Colleen. And I just want to make sure that you remember, don't neglect your symbionts. Don't neglect, the bacteria and microbes in the world around you. And good luck on the test.